Hello students, in this pandemic of COVID-19, stay at home, stay healthy and stay happy. We will try to reach and teach you through online classes till we are back on track. I have already taught you the general features and classification of hormones. Today I am going to teach you the hormone insulin. So what is insulin? It is a polypeptide hormone having 51 amino acids arranged in two polypeptide chains alpha and beta linked together by two disulfide bridges between 7th amino acid of alpha and 7th amino acid of beta and 20th amino acid of alpha and 19th amino acid of beta. It also contains an intra-chain disulfide bridge between amino acid residues of the alpha chain, and that is between 6th amino acid of alpha and 11th amino acid of alpha. Its plasma half-life is 4 to 5 minutes. The basal insulin secretory rate is about 1 unit per hour with total daily secretion of about 40 units. It is produced by beta cells of islet of Langerhans. The islet of Langerhans make up only about 1 to 2% of the total cells of pancreas. The pancreas lies mostly posterior to the stomach. It extends across the posterior abdominal wall from C-shaped concavity of duodenum on the right to the spleen on the left. The pancreas consists of a head, uncinate process, neck, body and tail. It is retroperitoneal except for a small part of the tail. This is the histology of pancreas, especially representing the islet of Langerhans and beta cell. The islet of Langerhans are ovoid, 76 to 175 micrometer, and it is a collection of cells. The islets are scattered throughout pancreas, although they are more plentiful in the tail than in the body and head. Humans have 1 to 2 millions of islets. Humans have 4 distinct cell types in these islets. These are alpha, beta, delta and F cells. They are divided on the basis of their staining properties and morphology. Alpha cells secrete glucagon, beta cells secrete insulin, delta cells secrete somatostatin and F cells secrete pancreatic polypeptide. Why it is called islets of Langerhans? A medical student, Paul Langerhans, was studying the structure of pancreas under the microscope when he noticed tissue clumps scattered throughout the pancreas. He named them as the islet of Langerhans. Now the structure and synthesis of insulin. Insulin is synthesized in the rough endoplasmic reticulum of the beta cells. It is then transported to the Golgi apparatus, where it is packaged into a membrane-bound granules. It is synthesized as pre-proinsulin. The gene for insulin is located on short arm of chromosome 11 in humans. Genes are transcribed to messenger RNA in the nucleus. After moving out into the cytoplasm, translation of the messenger RNA is initiated on the cytosolic ribosome with formation of an N-terminal hydrophobic signal sequence. This signal sequence aids in the transport of the messenger RNA and ribosomes to the rough endoplasmic reticulum. The N-terminal signal peptide penetrates the membrane of the rough endoplasmic reticulum. Further elongation directs the polypeptide chain into the lumen of the rough endoplasmic reticulum, resulting in the formation of pre-proinsulin. The signal peptide is cleaved and proinsulin is formed in the cisternal space. Proinsulin is now transported from rough endoplasmic reticulum to the Golgi complex, where it is cleaved, forming insulin and C-peptide, and both insulin and C-peptide are packaged in single secretory granules. So insulin is initially produced as pre-proinsulin, which is 100 amino acids long and which is cleaved in the endoplasmic reticulum, to proinsulin, which is 86 amino acids long, which is stored in secretory granules in the Golgi complex of beta cells. Proinsulin is further cleaved in Golgi apparatus by two calcium regulated endopeptidases, that is prohormone convertase 1 and prohormone convertase 2, rapidly followed by carboxypeptidase H to yield insulin and its peptide fragment, which is also called the C-peptide or connecting peptide. These both are then packaged in secretory vesicles and released when the stimulus arrives. So how this secretion, insulin secretion, when insulin secretion occurs? As glucose enters beta cells of islet of Langerhans by GLU2 transporters, it is phosphorylated and metabolized to pyruvate in the cytoplasm. The pyruvate enters the mitochondria and is metabolized via TCA or CA cycle. The ATP formed by oxidative phosphorylation of acetyl coenzyme A through TCA or citric acid cycle inhibits ATP sensitive potassium channels, reducing potassium efflux from beta cells. This depolarizes beta cells and calcium influx is increased. The calcium stimulates the release of insulin by exocytosis. The enzyme prohormone convertase 1 and 2 act on proinsulin to form the appropriate split proinsulins, and carboxypeptidase H removes the two exposed basic amino acid residues. Now, release of insulin. In healthy individual, insulin is secreted in pulsatile fashion, with glucose and insulin the main signals in the feedback loop. Glucose elicits the release of insulin from the pancreas in two phases. In phase 1, it begins 1 to 2 minutes after intravenous injection of glucose and ends within 10 minutes, illustrated by sharp spike representing the rapid release of stored insulin. Phase 2 begins at the point where first phase ends, depends on continuing insulin synthesis and release, and lasts until normoglycemia has been restored, usually within 60 to 120 minutes. So this is the graphical representation of release of insulin. Insulin peaks at meals, and there is a continuous basal insulin secretion with circadian fluctuation. So what are the factors affecting insulin secretion? Glucose, mannose, amino acids, gastrointestinal hormones as gastrin, secretin, cholecystokinin, gastrin-inhibitive peptide, beta keto acids, acetylcholine, cyclic AMP, glucagon, beta-adrenergic stimulators, theophylline and sulfonylureas. These are stimulators of insulin secretion. Whereas somatostatin, 2-deoxyglucose, alpha-adrenergic stimulators as epinephrine and norepinephrine, beta-adrenergic blockers as propranolol, digoxide, phenytoin, thiazide diuretics, microtubule inhibitors, and insulin itself are the inhibitors of insulin secretion. So what is the difference in range of insulin in human blood? After an overnight fasting, plasma insulin concentration in healthy, normal, non-obese people, it is 2 to 25 microinternational unit per ml. Concentrations up to 200 international unit per ml can be reached during a glucose tolerance test. Fasting plasma insulin values are higher in obese and non-diabetic people and lower in trained athletes. Rest of the insulin we will discuss in next class. Thanks.